So I'm very, very happy today to introduce to you Dr. Mats Persson and Dr. Daniel Johanson, both working at the Karolinska Institute. Mats has a background working with human monoclonal antibody engineering for many, many years. Worked at Karolinska Institute and Scripps Research Institute with these technologies. He has been very instrumental and supporting the foundation of this biotech company AAX, and I have to say that if you look at their names, you can also track the background on the foundation of this company. Daniel, also active at the Karolinska Institute, has several years of work in molecular immunology and also similar expertise to Mats. He is actually the in the lab inventor of the technology together with Mats Persson of the Seqitope and Opti-Mab platforms. So I would say from the horse's mouth we will get the information now about these technologies from Daniel and Mats. So please, I will stop sharing. You can share from your end now.
Right, thank you. See if I can share. Is it sharing?
Yes.
It's sharing, but not in presentation mode yet.
Okay, yeah. It's just that. No, everything was just super minimized. This worked so nicely just a few minutes ago. Let's see if I can do it again. Thanks. Still the same?
It's the same, but perhaps if you try to go slideshow, but you probably do that.
Let's see. Ah, terrific. No.
And then display and then display settings,
Yeah.
and you will stop.
Oh. It's like that. All right. Now we got it, I think. Okay. Great.
Yes. Excellent.
Yeah. Just a comment about the name there, AAX Biotech. Yes, it is our initials, but we made an after construction also. So it's Advanced Antibody Explorers, but you know, we had to come up with an alternative explanation. Oh, yeah.
Okay, yeah. Hello, everybody. He always takes any minute from my show. But anyway, I'm really happy to talk to you, and I'll give a very brief introduction. But I didn't even know how much you know about the antibody regeneration at DDD, but we do have large libraries, and from those we select new antibodies, and then there's a lot of work trying to characterize those different clones, right? And that, of course, depends on the target molecule, etc., and what we want to accomplish with the antibodies that are selected. So the very basic characterization first, so we get the amino acid sequence and binding characteristics. And also, somewhere along the line, we'd like to know whether the antibodies, when they bind, have a certain activity, if they are activating a cell, for instance, or killing a cell or whatever. And that's, of course, all dependent on the nature of the actual campaign for what target we want to find antibodies to. Now, next slide, please. Yes. One thing that we will talk about a lot today is epitope mapping, and that is where you try to identify as exact as possible the site for the antibody on the target molecule, right? And that has several benefits and important pieces of information. First of all, it's important for mechanism of action, and you'll see an example of that in Daniel's talk. And also, of course, it's also another way of distinguishing between different antibodies. And all this data are important for regulatory issues and also for patent purposes. So more and more epitope mapping, and of course, we're sort of selling the analysis, so we think it's very important, but I think also the field is getting interest into this. And as you may know, there's also a lot of work trying to use AI to in any way try to assess or at least get some type of basic assumption what the epitope will be for a new antibody. And maybe Daniel will talk more about that. Next slide, please. Now, for epitope mapping, there are some very quick and sort of dirty ways to do it. Peptide scanning is quite popular, where you split your target, if it's a protein, into small peptides. And of course, then you get a little bit of information whether the antibody binds to a linear epitope, as we say, but it won't cover conformational epitopes. Another way could be antigen fragment analysis, in particular if you have known domains of a large protein. That could be useful, at least to understand that you want to hit this or that domain. Most often, antibodies are more, let's say, precise than that. So even just hitting a different domain usually doesn't do the trick. It has to be on a certain position on that fragment, sort of. Anyway, so if you get down to more precise methods, they're usually quite cumbersome. X-ray crystallography has been the golden standard for many years, but it takes a lot of effort, and not only producing a lot of protein, but also the machinery, the knowledge in the people who are doing it, etc. HDX-MS is slightly different and used a lot. It's has a sort of air of being cost efficient, and we can talk about that a lot. And now, more recently, cryo-EM has sort of been introduced, and is very popular too. And I'd say X-ray crystallography and cryo-EM are hopefully very precise. HDX-MS are not really, but if we take the next slide, please. HDX was a method that we used a lot at DDD and still do, but for some projects it doesn't really work. And I personally was PI for a project at DDD where this happened. So it wasn't successful at all, and partly because the target protein for the antibody was very hard to express in the first place. So we got it only in fairly low concentrations, and somehow it just disappeared when we wanted to isolate it, etc. So in the HDX-MS process. And I have a figure there in the middle. HDX-MS really, sort of the basic principle is that there are certain parts of the protein which are accessible for exchange between ordinary hydrogen and deuterium. And so if you, obviously the ones that are possible to exchange with deuterium are the ones that are not covered either by another chain of the own protein, or by an antibody coming and blocking sort of the access to the solvent. And you can do that, and then you chop it up and you determine whether there are deuterium or hydrogen in the various amides. But for this protein, it just didn't work, and we tried it several times. And the people who did the HDXMS analysis were of course, very talented and a lot of experience. But in this case, it just didn't work. And that's when Daniel, actually, who was working in my lab at the time, came up with a new solution, and that's what brought us to Seqitope, and you will hear a lot about the details there. But you will also hear about something that I just present on the next slide, please, and that is single-chain Fv antibodies. You might have heard about it, that it's really a small, I don't know if you can point.
Let's see.
It's a small part of of the antibody molecule, which is the actual most important one, the one that actually creates the binding to whatever target this antibody has, right? And it was discovered by Jim Houston in the late 80s, and he, next slide will be there. He put a peptide linker between the two different protein domains. And this was very practical in a way, because you can play around with it a lot. It's a single cistronic construct, so it's been very, very fruitful for protein engineering when it comes to antibody constructs of various kinds. I think, okay. So it's been used a lot, for instance, for creating bispecific antibodies. So here's the single chain added on to the light chain in this case. And also more recently as sort of the specific molecule for CAR T cells that recognizes a cancer cell. And then you express the single chain on the surface, obviously, and then with a lot of signaling machinery down to the T cell here. Now, as Daniel will also talk about, if this aggregates on the T cell surface, the T cell becomes activated even without having a cancer cell in the neighborhood. And that's a big problem with this single chain constructs, that they do aggregate a lot. And that's partly because of the CDR regions they have, I mean the specific binding sites of the antibody, but also in some cases a more general feature. And that's another problem that's been hunting the field for a long time, in particular in CAR T cell, I'd say, but also when it comes to different single chain constructs. And that's another thing that we struggle with also in our lab. But fortunately, Daniel came also to rescue there. So I think that's what I'd like to say about this, and you can take over.
And I mean, we ran into these two problems. Both kind of was related to this DDD project, where we tried also bispecifics with the DDD project, and you know, to make it work better, we had to find a solution for single-chain FVs. And these two solutions actually, you know, we turned those into a new company. So we're called AAX Biotech. And so we're centered around these two technologies. Seqitope, which is this epitope mapping solution that Mats mentioned, and Opti-Mab is the new format for single-chain FVs. So I'll talk a little bit about these two technologies. So first, Seqitope, which is a high-throughput, high-resolution epitope mapping technology. So yeah, and what is epitope mapping? Mats covered that, right? And Seqitope is based on massive parallel mutagenic scanning. So I mean, like, people have tried similar things, and I'll get back to why, you know, Seqitope has some unique angles there. When we have high throughput, right now we can run, well, comfortably we can run 40 antibodies in parallel, and most other methods are, you know, one at a time. And we also have high resolution, so we get a signal from every single amino acid, how much it contributes to binding. And we have applied for a patent for this. So again, I mean, now you talked a little bit about this, Mats, but if comparing to other methods, cryo-EM co-crystallography, that's a structural analysis, and HDX-MS is based on spherical blocking. And all of these methods basically show what amino acids are proximal to the antibody. And then, of course, you can add layers of interpretation there or biophysical models to say, okay, here's a salt bridge or hydrogen bonds, etc. But in general, proximity is not the same as contributing to binding. So the information you get is something like this. You know, these amino acids marked in blue are close to the antibody, but it's not always easy to say which one actually are involved or contribute to binding energy. Compared to Seqitope then, we get a signal from each single amino acid. So in our case, we get information looking like this. So we actually get a signal from each amino acid. And even though an amino acid is very close to the antibody, we do not get a signal from that one if it's not involved in binding. So that's kind of an upside. The downside is that we only get data for the antigen. I mean, like in a crystal, you see both of them, so that's super valuable, right? You can also tell things about the paratope and so on, but unfortunately, this is not the case. So that's kind of a drawback with Seqitope. Of course, we can flip the system around and do a paratope mapping, but that's kind of a separate experiment in that case. And again, I mean, like this, just comparing to some other methods. In general, we can say that the unique things is that we're working with high resolution, high throughput, and we can also resolve complex epitope, that is discontinuous epitopes. And that makes it pretty unique. I can also mention that now we're putting the feasibility as high here. So the vast majority of our customers have actually tried other methods. So they come to us when they have failed, typically with cryo-EM or HDX-MS. Those are the two common ones that they've tried, and it has not worked, and then they come to us. So and we've been able to solve their sort of epitope mapping. So that's why we say, okay, maybe we're a bit more feasible. But with that said, it's a prokaryotic system. And it's also based on solution. So if you have multi-membrane spanning protein, that's a big challenge. We have worked with that, but then we're typically working with ectodomains or loops for multi-membrane spanning proteins. So there's some drawbacks there as well. So here's our first proof of concept study. So this actually comes from the DDD project. In this case, it's a published antibody. So the image to the right, that's the epitope as shown in the publication. And that was made by partial alanine scanning, which is a lot of work to do alanine scanning. Basically, you make a mutant of each amino position and then produce it, purify it, and then do binding studies. So when we ran Seqitope, we got a very similar epitope, and we also found some other positions that are probably involved in the binding, including the ones that they in the published paper. We also get an intensity of each amino acid, so how important it is, not just a binary yes or no, but rather, and I'll show you, I'll show you in a later slide. Something that makes Seqitope a bit unique as compared to other methods when you do deep mutational scanning is that we can differentiate if an amino acid is structurally important or important for the epitope. Because this is like one of the main problems when it comes to mutagenic scanning. So if you introduce the mutation and then your antibody does not bind, it could mean that you have mutated the epitope. But it could also mean that you mutated something that is very important for the structure and the whole protein misfolds and then your antibody does not bind. But of course, that's not the epitope. So you have to find a way of differentiate between these two cases. And this is a bit of a sort of our secret sauce. But I can just show you some general ways that other people have tried. you can, for example, look at the surface accessibility score. If you have a structure or crystal or something of your protein, then you can see, okay, everything that is not on the surface cannot be involved in the epitope, or typically not, at least. So then you can kind of filter those out. So that's one way of doing it. But then you need a structure, plus it is a kind of an artificial in silico filtering. In this case for Seqitope, we're actually doing a separate wet lab step. So it's like a parallel experiment that we run side by side with the epitope mapping. And we kind of get a data set which we can use to normalize the epitope mapping data set. So that kind of it nullifies the signal we get from structurally important residues. And I'll just show you some data. So the image to the left, that is, if we just look at the raw data that we get out of a mutational scanning, it looks like that. And it's very difficult to see. I mean, like, there's a lot of residues inside and it's all spread out. Maybe you can see that the epitope is kind of here, but it's very difficult. So, but when we run this control to normalize the data set, so we remove the contribution of all the structurally important ones, it looks like this. So we get a much clearer image, and this is the published epitope as a comparison. And this is just another data set. This one will look pretty good, but it's still some amino acids up here without this extra step, and you get a clearer image again with the control. And this is the published epitope in this case. So a case study, just to show you some more data. We were working with Oblique Therapeutics. They had antibodies targeting TRPV1. So they had 3 antibodies, and they've done some epitope mapping. And according to that epitope mapping, they all had the same epitope. Still, 2 of the antibodies had an agonistic effect, and the other one an antagonistic effect. And they couldn't understand it, because according to the epitope data they had, it looked the same. So we helped them out. Now this data is kind of a bit of anonymized. So we have removed what amino acid it is. Now it's just like some positions here. So, but then we can see that antibody 1 and 2 and 3, yes, they have an overlapping epitope pair in these amino acids. Sorry, it's something with the monitor here happened. What to do? Yeah, eventually can you see them? Yeah, it says like the monitor will shut down in 37 seconds. Exciting. But anyway, but still with the high resolution data we get, we could actually see that antibody 1, 2, and 3 actually had different epitopes. And here's a table with the actual raw data. So you can see that this BC, that's the binding contribution score. So you can see, okay, amino acid position 14 is actually very important for this interaction, while amino acid position 16 seemed to contribute a bit. Of course, you're getting into some kind of, well, is this also involved or not, and so on. So, but we can say as much as 16 is not as important as 14 in this case. So we could actually, with the Seqitope, resolve it in much higher resolution and also answer that question: why did they have different pharmacologies? And we were not aware of this at all when we started the project. This is something they told us afterwards. So another study. This is a pretty interesting thing. So we believe we can also use Seqitope for affinity maturation. This is a little bit odd because now we're working on the antigen side, and typically, of course, you want to affinity maturate the antibody. But this is just like a proof of concept study. So we started off with another kind of proof of concept study. So this is the RBD domain of SARS-CoV-2. And this epitope was determined by cryo-EM, and you have the residues here. And then we ran it by using Seqitope. And then, again, we found the important ones, but this Q493, that didn't show up in the Seqitope analysis. So we were wondering, okay, what's going on? So this is actually the data for this data set, and it's a discontinuous epitope. So here's the cutoff of 0.5. So all of these are, well, the amino acid you can see in this table. But then looking at this Q493, where did that show up? Here with a very negative score. And we didn't understand. We didn't expect anything like this to have a lot of negative scores. So we were like, okay, what's going on here? We expected either score close to 0 or something high, which would indicate an epitope. So we looked into this and also the other residues that had a very negative score. And we could see that all of those were located in the kind of epitope area. So the hypothesis was like, okay, could these positions be suitable for affinity maturation? And you know, when we introduce mutations into our sort of library, we introduce a lot of different mutations, not just alanine, but many different mutations. And when we looked into these specific positions, we could see that non-wild type amino acids had been enriched. So for example, in this case, it was a 11 times enrichment of tyrosine in this position, for example, which was very interesting. So we actually made these mutants, produced them. So one where we mutated this Q493, and then another one, a combination of these two mutations. So which are the top two ones that Seqitope kind of suggests to be affinity maturating. We ran an experiment then with some controls. This Ty1, that is the antibody that we're investigating. The epitope is marked in green here. And then as two controls, we had one which we called Mab dis, as in distal, because that epitope is distal from this area. So if we introduce mutations here, this binding should not be affected, right? And then we had another overlapping antibody that bound straight into this area. And in this case, Seqitope pointed out that Q493 is very important for this OVR antibody. And then we mutated these two positions, which are the 450 and 455, the two most enriched of non-wild type amino acids. So this is the data, and this was actually performed by the guys at DDD. Oscar helped me out running the SPR. And again, looking at this distal one, you know, the two mutants as compared to wild type, not much of a difference. So that makes sense. And then we had this one with an overlapping epitope. And if you mutate this R, which is key for this epitope, you see that the binding is totally, there's no binding. It's the lower curve here. While this YQ mutant, well, if it affects it a little bit, which could make sense since it's in the middle of the epitope binding. But the interesting thing was for Ty1. Here we could see the wild type is this curve, and the R mutant is this curve, and the YQ mutants, I mean, the two top suggestions from Seqitope, so to speak, for affinity maturation resulted in this curve. So very much improved binding. And if we look at the affinity, KD improvement versus wild type, actually, this should, it's the wrong names here, but this is the distal one. So they all have the same. The OVR one, which is affected if you mutate this Q493. And then we have the Ty1. So if we follow the kind of the input from Seqitope, we get a very much improved affinity. So to summarize Seqitope, we can solve complex epitopes. We have single amino acid resolution. It's high throughput. We don't need any antigen as compared to many other methods, since we produce the antigen as part of the method. And we only need tiny amounts of antibodies. So typically we ask for 10 micrograms. We could work with less also. And just some other areas where which we explored with Seqitope. So monoclonal epitope mapping, that's what it was developed to do. We could also do paratope mapping and, at the same time, you can hopefully will get some information on how to affinity maturate your antibody. And then basically any protein-protein interaction, it doesn't matter really what kind of protein it is. We are investigating polyclonal mapping in a collaboration. And a small molecule, we're talking to a company if we should do this. We haven't tried it, but this was something that we want to explore. And then AI, we produce very detailed data sets. So we have worked with a German company there, proof of concept that the Seqitope data is really contributing to better outcomes in an AI machine learning setting. So over to Opti-Mab. And Mats already had an introduction there about single chains and how they tend to misbehave. And Opti-Mab is really, now it says general applicable molecular stabilization strategy, but now we can talk about it. We have it in the patent application. So we have a new linker sequence that is stabilizing the interaction between VH and VL. And what we can see is improved the production levels. This is in a collaboration with the CAR-T team. They had a problem with two of their single chains, so single chain 1 and 2. In the standard format, they were horrible actually to produce. And we just swapped out the linker to the Opti-Mab linker, and we got very much improved. This is pretty extreme, I would say. This is typically we see between 4 and 8 times. That is what we typically see. And this was a clone that already expressed very well, but still that improved the production level by 2 times. We also have data on that we improved the stability of this one as well. Looking at DLS, just to see aggregates, this was one of the most difficult single chains that we worked with. And basically after purification, all of it was in aggregates. But if you produce it in the Opti-Mab format, you get it as monomers. And here's some SEQ-HPLC just basically showing the same thing. This is a human one. In standard format, it looks like this. This is where the single chain peak is supposed to be. So it's like basically nothing. And when we produce this exact same one, just swapping out the linker, it looks like this with the correct peak here. And here's another one. This is a mouse. So we can work with both human and mouse. We have not that much data on mouse, but at least for a few. So it's very broadly applicable, this linker solution. This was very horrible in the standard format, but in the Opti-Mab format, pretty good. So a case study. We have applied this to an FDA-approved drug that has been on the market since 2015 called Blincyto. Blinatumomab is kind of the scientific name. So it is consisting of 2 single chains like this, one targeting CD3 and one targeting CD19. And we produced it in the standard format as described in the patents. And then we also made an Opti-Mab version of it and produced it in CHO cells, or rather the CRO did it for us. We checked for stability, accelerated deterioration, 40 degrees for 1 week, and quantified loss of soluble protein. And then also made sure that it still did its job, sort of in a redirected T cell cytotoxicity assay. Basically see that, yes, it recruits T cells to kill CD19 positive cells. So here's the data. So the improvement was not that high when it comes to production, but one should remember that this is a therapeutically approved one, and they probably put a lot of work into kind of maximizing the production. So it's probably more difficult to improve it further. This is the loss of soluble protein. So in the kind of the commercial formulation, it lost 16%. After a week at 40 degrees, while in Opti-Mab format, only 2%. And this is the RTCC assay, basically showing the efficacy of the drug. And you can see that, yes, they have the same, maybe even in Opti-Mab format, you have like some improvements here. But in this, it looks very, very similar. So to conclude, we see all these improved functionalities. I know time is running out also, so I'll push through here. And we also have some data with CAR-T unfortunately, we cannot show this. This will hopefully be published now during the spring. So we can see decreased CAR aggregation and a lot of other positive markers in the T cell. So to sum up, superior expression, reduced aggregation, improved stability, and we see retained functionality in binding. And yeah, we can see improved CAR-T properties. And we have also applied Opti-Mab to on-market bispecific antibody and improved those properties. Just a few words of AAX Biotech. These are some of the collaborators and customers that we've had that we can talk about. We have a few other ones, but Daiichi Sankyo, that's the big one, now working with us in the Opti-Mab sort of field. And yeah, we also won the Golden Tiger at Scandinavia, so that's great. Yeah, and that was the last slide. So thank you.
Thank you very much, Daniel and Mats. So we are open for questions, and it was really interesting presentation.